Hi, I'm delighted to introduce Shin. Shin received his PhD at the University of Georgia, studying fundamental mechanisms of T cell biology, and then joined the lab of Ilyan Ilyev in Cornell and helped develop an entirely new field of the mycobiome. So rather than focus on the bacteria that reside in the gut, focus on fungi and how they shape innate and adaptive immune responses. This led to kind of seminal publications, first author papers in Cell-Hosted Microbe in Nature, and the receipt of a K99R00 from NIAID. So Jin, we were thrilled to recruit him in early of 2023, and he will continue to study how fungi kind of communicate with the host and other commensal microbes and dictate innate and adaptive immune responses in the context of autoimmunity and anti-tumor immunity. So Jin.
It's my pleasure to give the talk here. Thank you so much, Andy, for the introduction. So our lab is really interesting for the host and microbe interaction. Ralph also gave a little bit of introduction about the fungi and also the plants. We also started for the fungal species that also be able to interact with the human immune system. That is really exciting and interesting for us. Give a little bit of introduction for the fungi. We know fungal is well known to cause the infection with 100 million infection cases reported every year. 1.5 million people die of the fungal infection. We know there are pathogens that are capable of causing the infections. However, one important question is, are they live in the healthy human individuals? This is a question was a lot of scientists try to address this kind of question. The first case of the fungal infection was described by one of the most famous physicians in history, Hippocrates. This is like a first describe of the fungal infection in the oral. However, they do not know what kind of the organism caused this. So until 100 years ago, when scientists called Harry Anderson, he started to address this kind of question, whether the fungal species be able to live in the healthy individual. So because the technical advance, the microscope, I think every microbiology lab those, they be able to use the microscope to look at for the culture-based organism, they be able to find the yeast-like fungal species in the gut of the healthy individuals. This data suggests like a fungal species probably be able to live in the healthy individuals. The last decade, I think everyone knows the gut bacterial community became really important scientific topic. So this also triggered a group of scientists to address this kind of question again, to re-ask whether fungi be able to live in the human gut. So this is some of the data came from my previous mentor, Dr. Ilyan Ilyev. He started ask this kind of questions in 2005, and then he developed a technique to study for the fungal species. What he utilized one of the soluble Dectin-1 probe. Dectin-1 is the receptor of the beta-glucan. Beta-glucan is the cell wall component of the fungal species. In this studying method, he be able to locate for the fungal species living in the mucosa area. This kind of data is, it's not very clean because the beta-glucan probably also express about some other food junk, everything. But still, this is the first evidence to demonstrate that living in the very tiny of the mucosa area. So with another technique of the next generation sequencing, so a lot of scientists actually started to use the next generation sequencing to address those kind of questions. They find for the healthy individuals, there's a diverse for the fungal species living in the gut. With 13 of the conditions, such as antibiotic treatment, immune suppression therapy, transplantation, or mucosal damage, this be able to promote for the fungal expansion in the intestine. We call this the fungal dysbiosis. Fungal dysbiosis now has been associated with many inflammatory diseases, including one of the disease Libras just described the inflammatory bowel disease, also the allergic area inflammation, and also the liver disease as well. Several facts about the gut fungi, what we try to highlight. They are less complex when compared to the bacterial community, but they're highly immunogenic. I hope I have a few of the evidence to convincing you they're really immunogenic, be able to remember by the host for a super long time. This came from an immunologist perspective. So the first evidence I want to show for the fungi and the inflammatory bowel disease. So for the inflammatory bowel disease, the one type of the chronic intestinal inflammation. So fungal antibody is one of the serological biomarkers for the patient with Crohn's disease. This is a subset of the inflammatory bowel disease. And also, this is highly associated with the disease severity. So what is ASK antibody? ASK antibody is the one of the antibody targeted on the fungal cell wall components. This is a fungal cell wall. This is a menin. This is the target of the menin. So this is the first piece of evidence coming from the human. The second piece of evidence came from a human. So one elegant study was from a German group led by Patrick Becker. She's an immunologist focused on human immunology. I think she did a fantastic work for this. What she did, she sorted out CD4 positive memory T cell from the blood of the healthy individuals or the patient with the allergic area inflammation. And then she stimulates, this is a memory CD4 T cell from a human with a variety of the microbial lysate, and then measure for the cytokine production. Here, what you can see, one of the important cytokine called IL-17 be able to pop up within the CD4 T cell population. And it's very unique to the fungal lysate, not for the bacteria or the other fungal species. This is very unique to the Candida albicans, one of the human fungal commensals. So they also further to show the Candida reactive memory CD4 T cell contribute to the lung inflammations. So I like to show a little bit more piece of the work to demonstrate commensal fungi are really important to contribute for the human disease. But for our lab, we really interesting to understand how the host communicate with the fungal species. What kind of fungal factors to be able to recognize and then initiate for the immune responses. So there's three different components. Now what we know, one is the glycan, another is a protein that be able to initiate for the antifungal immunity. So in 2001, one of the classic paper was published by the Gordon Brown. He cloned and also described the first dectin receptors. The first receptor receptor called the dectin-1. This is binding to the beta-glucan. It's be able to initiate for the immune signaling cascade, activate C-carnine signaling, initiate AFK-B signaling pathway. For the latter, so a lot of scientists identify other C-lectin receptors as well. So one of our recent work, we demonstrate a single protein peptide. This is a perform protein called the candidialysin that be able to initiate for the tissue inflammation. Candidialysin can activate for the NRP3, depending on the inflammation activation for the macrophages, be able to produce for the R1 beta, and then further to control for the T cell immune responses. However, we still do not know how they initiated for the epithelial cells inflammation activation as well. So another question is remain unknown. I think we also work on this at UT Southwestern. whether the fungal nucleic acid be able to recognize by the host immune responses. I like to share some data about our previous work. So Candida albicans is a human commensal. They peacefully live in the human gut, but they have two different forms. So one is ester form, another is hyphae form. When they living as ester form, they are healthy. They do not produce valence factors. But when they switch from ester form to the hyphae form, they started to produce a lot of hyphae form associated valence factor. One of the key factors called the candida lysine. Candida lysine was discovered in 2016 by our collaborators, Julian Neckleg and Hubie's group. So they saw candida lysine be able to induce for the cell damage. So we define the candida lysine's function in the intestine, how they communicate with adaptive immune responses. What I did, I collect a few of the fungal strain. This is the Candida albicans clinical isolate from a human. We use genetic editing, the CRISPR-Cas-based system to delete the candida lysine. Then look at it for the cell damaging. You can see here, this is the high damaging strain. It means that produce more candida lysine. If you delete candida lysine, they fully lost the capacity to induce the cell damaging. So this is the low damaging strain produce less candida lysine. If you delete for the candida lysine, so they lost the capacity to induce cell damage as well. This data indeed indicate candida lysine is really important for Candida albicans to induce the cell damage. What about for the in vivo immune responses? I'm a T-cell biologist. I'm really interested in how this protein peptide control adaptive immune responses. Then we look at one of subset of immune cells, TH17 cells. TH17 cell is CD4 T cells express an inflammatory cytokine called the IL-17. There's a classic responses to antifungal immune responses. So this is a high damaging strain. When you colonize them in the germ-free, this is the environment really clean. So they induce really strong TH17 cells infiltration in the colon. However, if you look at it for the candida lysine knockout strain, so the TH17 almost 19 to 80% TH17 was gone. So this is similar with the low damaging strain. If you knock out the candida lysine from a low damaging strain, so the phenotype look like didn't really change it too much. This data suggests candida lysine is really important to induce for the TH17 mediated immune responses. We also further recapitulated the data from one of the mouse model, DSS-induced Cladder's model. This data also demonstrate candida lysine is really important factor to induce for the immune responses. So we show a secret protein peptide from the fungal commensal be able to activate for the tissue immune responses. What about the nucleic acids? Why we address those kind of questions? Because it look like very obvious nucleic acid be able to induce type 1 interferon production. Their bacterial RNA and the virus RNA that are inflammatory. So one of the questions is eukaryotic organism, their RNA has been extensively modified. So like mRNA has the capping system and chemical modification and RNA editing that remain as non-stimulatory. So that is true for the majority of them. And for the DNA recognition, we know DNA probably be able to translocate into the immune cell cytosol active cGAS-STING signaling pathway. However, fungi remain as extracellular organism. It's really hard for them to deliver their DNA inside of the immune cells. So that just became a question for us to address whether nucleic acid be able to trigger immune responses. So we expanded this kind of work when we moved to UT Southwestern. I think that is quite excited. If I have more time, I like to share a little bit more. So we found one of the unique RNA that expressed in the ribosome, but it's only came out on the polysome. That's very interesting for us. We name it called the FSRNA. So we further collaborate with James Chin's group. We find it goes through mitochondrial antiviral signaling pathway. It's be able to recognize by one of the cell signaling pathway. This is be able to utilize detect for the virus. That is quite exciting for us. I like to thank a few people in our lab, and also I want to thank a lot of collaborators. When I moved to UT Southwestern, I almost like borrow everything from a lot of scientists here. The environment is really supportive. I want to thank Annie Cole, and also the Yannis group for cell line, and also the James for the mice, and also the cell line as well. And we did the ribosome isolation work with Jim Fang. I'm so lucky to have some iron biologists that's really close to our lab. And also about a lot of people when I moved to here, to John, and also Lisa for the cell line. I think a lot of people, they are often the insight to us. Thank you so much. Yeah, I'm happy to address any topic.